Today I'm going to be sharing the details of our very cool project another one that we did with Jana Gevertz on mitigating non-genetic resistance to checkpoint inhibition based on multiple states of immune cell exhaustion. So first, a bit of history on immunotherapy, and until about, I guess, as not far ago as 30 years ago, the mainstream thinking was that cancer cells are too similar to normal cells and immune cells just can't recognize them. When I was starting to learn about cancer in the early aughts, this was still the paradigm that I learned. So it's really not that, the shift has not been that old. And then we learned that immune cells actually have both brakes and accelerators to both fight pathogens and avoid autoimmunity. Because, of course, the immune system has this dual role. It needs to clear invaders, but it also needs to make sure it's tampered down and not kill its own cells. So we found that cytokines can activate immune cells like an extra gas pedal. There's, for example, an example of IL-2 therapy from the early aughts, which worked in some cases, but it had limited efficacy and was very, very toxic. It was really sending immune cells into overdrive. Why didn't it work overall, even despite the toxicity concerns? Well, the thinking is that we still kept the other foot on the brake while pressing on the gas. So you are on one hand trying to press accelerator, on the other one is still keeping the foot on the brake. And it's not until we found these extra brakes, these extra brake pedals, the checkpoints, that we actually started seeing immunotherapy work. And again, this is not that long ago. The first checkpoint inhibitor was approved in 2011, as far as I remember. By the way, if you want a little bit more history on this, I really recommend The Breakthrough by Charles Graeber. It goes through history of immunotherapy and this thinking and the culture wars and dogma wars through starting from the 19th century and the colitoxins. It's fascinating. Well, so as remarkable as immunotherapy is, and specifically checkpoint inhibition, it only works in a limited number of cases. Some estimates say 20%, could be a little bit more, a little bit less. And my interest here is to try to think through why. So kind of a bit of a background, just in case, on how immune checkpoint inhibitors work. So normally you have your T cells and you have your immune cells and you have these molecules. So PD-1 is an example of a checkpoint. And T cells, they express these molecules to really sample other cells and check, are you to be targeted or not? Are you safe or should you be killed? So when the cell signals, so PD-1 is a checkpoint, if it binds to a native ligand like a PD-L1 ligand on a normal cell, or in this case on a tumor cell, it gets a signal, do not kill. Tumor cells express these PD-L1s, so T cells don't kill them. And the term that has been used for these T cells that are supposedly alive and can do things but are not actually killing, not really killing, they've been called exhausted. Well, and the way that these checkpoint inhibitors work is that you essentially try to break this native signaling. So the antibodies outcompete the native ligands. So the T cell now cannot bind to this and get the signal from the tumor cell that do not kill me. So then the killing can actually take place. Fantastic. Now, we used to think that this exhaustion state is binary. So you basically have exhausted cells versus not exhausted cells. But there actually are, and we're kind of learning more and more about it, that there are actually multiple states of T cell exhaustion. And only some are susceptible to checkpoint inhibition. Here I'm referring to them as reversibly exhausted immune cells. There are different classifications, but for our purposes, functionally, this is good enough terminology. Of course, when I see something like this, I immediately put on my evolutionary biology hat and think, well, why did this evolve in the first place? And this is something we also started talking with Joel Brown, like another colleague of mine with whom he's an ecologist with whom we've been discussing, and always exciting to talk through different evolutionary perspectives here and looking for ideas. Do we see that in nature as well, and why that would have happened? We have, I think, like a little review paper perspective on this as well, if anybody's interested. But for this particular case, So what we now are learning that subsets of exhaustion are actually characteristic of chronic viral infections. And it seems like a lot of these characteristics are actually very similar to the exhaustion phenotypes that we see in cancer. So this whole multiple stage of exhaustion likely evolved as a mechanism of protection from autoimmunity. Again, cancer does not invent anything. Most of the adaptations seem to be something that is used, that exists, that has evolved for a purpose, but then gets activated or used either out of proportion or out of context. And this one is almost as if tumors are, quote unquote, pretending to be chronic infections, and therefore saying, no, immune system, calm down, because it could be less damaging for the organism to coexist with an infection than to fight it to elimination. And which is where this balance of autoimmunity versus cytotoxicity kicks in. Okay, well, if this is the case, if we assume that this evolved to protect the person from autoimmunity, then how would these cells know? What would be the triggers for transitions through these different stages of exhaustion in chronic infection? And could they potentially be the same in cancer? So this was kind of the thinking here. And as I'm also thinking through this, well, how is the immune system communicating with itself, with the different constituents in the cells? Really, we have inflammatory response and inflammation. And so my hypothesis here, or the hypothesis that we were discovering, assessing here, is that maybe it's something that must arise from as a function of this immune disease interaction. Possibly like a strong inflammatory response, which is a hypothesis we took forward here. Could be prolonged antigen stimulation. That is another paradigm and another hypothesis on what triggers this, or a combination thereof, but something that is triggering these transitions. So could this lead to emergence of resistance to checkpoint inhibitors? Well, let's look at the framework that we're looking at here. Like in a world where this is true, you would have your tumor that appears, and the immune system starts to react. So you have cytotoxic effector cells, and if everything goes well, they might be able to eliminate the tumor, and then there's no story, no problem to solve. Everything is wonderful. But if this keeps going, maybe some of these cells start transition to this reversibly exhausted phenotype. So what is the function of this? Normally, that's how I think about it, is that the immune system goes in, just guns blazing, trying to kill everything. At some point, there must be a signal that you're starting to get too many civilians, too many of the normal cells. So maybe it makes sense to slow down. And the hypothesis is that, okay, then maybe the reversibly exhausted phenotype is when they're starting to basically sample different cells that they are attacking, saying, okay, are you friend or foe? So this is a way to slow down the immune response without completely shutting it down. Well, if this keeps going, maybe you have something like, so inflammation might be increasing because you have this ongoing battle, and maybe there is some kind of a transition threshold between the cytotoxic, just guns blazing state and the reversibly exhausted T cell state. So a threshold of, assuming here, inflammation, however that is quantified. This is kind of right now a relatively generic quantity, but something that slows down. Okay, maybe I should slow down and look at things. If nevertheless this keeps going and the inflammatory response is really, really high, we're hypothesizing there maybe is another threshold of transition from reversibly to terminal exhaustion. And the idea here would be, okay, so it's interesting, the terminally exhausted cells actually are cytotoxic. They can kill things, but they are very short-lived and they just kind of kill and die. Reversibly exhausted T cells are not actually cytotoxic. They are not, and there were experiments, there's one from the paper by Brian Miller, for example, showing that this class of cells, it is targetable by checkpoint inhibition, but it actually is not cytotoxic. These guys are not targetable, but are cytotoxic. And that really is what creates this interesting dynamic that we're going to be discussing and showing here. So the hypothesis is that increase in inflammation, or something that is triggered by these interactions, causes the cells to transition, T cells to transition between these states of exhaustion. And we assume, this is hypothesized, that there exist thresholds that signal these transitions. Here, just doing for inflammation and assuming the quantities there, threshold F1 and F2. So now let's think through this. If this is what is happening, the drugs, so checkpoint inhibitors, take these Y2 cells, the reversible exhausted T cells, and basically put them into the cytotoxic bucket. But if the battle is going, and so now they are now ready to kill more things, well, this keeps increasing. So as these cells are being put back in here, if this is keep happening and the other systemic or probably local inflammation passes the threshold, then what you have is they're not staying in this. So they might be transitioning to terminal exhaustion. So at some point you might find yourself that yes, you're given the drug, but because this is happening, the cells essentially are skipping or moving really quickly through these states and end up being terminally exhausted. So yes, you keep giving checkpoint inhibition, and it's not working because you've depleted the pool of the reversibly exhausted cells because the underlying reason for this transition is not addressed. So that is the idea behind this framework and this theory, and this is the implications of this is what we are going to be discussing here. And the question was here, okay, if this is what is happening, if this is this mechanism of resistance, essentially, but it's not caused by genetic resistance because you're not killing off sensitive cells. You are not, but you are creating an environment that does not favor targetable cells. So is there something you can do to mitigate this? For example, through dosing regimen. So we created a relatively simple, like conceptual PK/PD model. The PK, so drug pharmacokinetics, which is tracking drug concentration over time, is based on mouse anti-PD-1 drug. Got it from the literature. The dose-response relationship, so the impact of drug concentration on tumor growth reduction, is not parameterized to real data. Didn't really have data on that, but still, this was good for the exercise we wanted to do here. So we have just a control that, okay, we have our no treatment and there's some carrying capacity, or the immune system is kind of keeping some track of things. But basically, this is our kind of simulated tumor without treatment. Then we gave 25 doses, arbitrarily chosen number, every 6 days versus every 7 days. The idea, the hypothesis was that maybe you need to let the information clear out a little bit more before, or not clear out, but there is this dynamic of this, whatever is triggering this transition, that needs time to act in order to be able to see the effects. And so we had the simulation, like, okay, here, for example, the same dose was given every 6 days, and you have complete tumor elimination, versus every 7 days, same everything, but, okay, so you have, oh, it works, but, okay, it didn't catch up, oh, it didn't catch up, oh, and now the more cycles you give, kind of you see less and less response over time within this framework, and then eventually it just stops responding at all. Interesting. Why would that make such a difference? Again, have my kind of ideas and hypotheses on this, but it is a model. We can explore everything. So we decided to look at, kind of do a more systemic analysis or systemic study of different dosing strategies. And to evaluate for any patterns, we created this dose fractionation sweep. So we selected some total dose, and then we would fix the number of doses, so subdivide then the number of doses of, like, from the total dose. And then we looked at if changing the dose amount and spacing between doses will have impact. And this is kind of what we saw. So red is kind of treatment failure, so tumor growth. Green is success, tumor elimination. And we are seeing some patterns here. So it looks like one way, so remember this is a total dose, and then the question is how you divide it up, which is the number of doses. So if you divide it into smaller pieces, and each dose will be larger. So this is large doses to smaller doses, and spacing is obviously is self-explanatory. So here we have large doses that are given very spaced apart. So it can work. And this is the high dose, low frequency strategy. It's typical, like maximum tolerated dose, like, or more typical protocol. Here we have this intermediate, however one defines it, kind of relative to the different ends. So you have larger, maybe smaller doses and smaller spacing between them, but that can be a strategy. And then finally, we have this region where you would have low dose, high frequency. So it's metronomic-like therapy, where you have a lot of small doses, but they're given closer together. Interesting. So now that we looked at this, but we wanted to see, but why, like, what happens at these boundaries? Why does one day make such a difference, up or down? Again, this is a model. We can do anything with it. So we were looking at the individual trajectories. This is a randomly selected dose, for example, of 3. And so given every 4 days, every 3 days, or every 1 day. These are not real protocols. This is for simulation. But still, we're looking for patterns right now. So we see, okay, so this one, tumor kind of escapes growth. This one, it was eliminated. And this one, again, was not. Why? So first, we wanted to look at the relative proportions of different immune cell states. So is it pure effector state? Is it reversibly or terminally exhausted? And as you can see in both cases, so this is the top panel and the bottom panel, the escape panel, so the ones where it didn't work, the kind of dominating proportion of cells is my Y3 is my terminally exhausted cells, which intuitively makes sense that, yeah, they are the ones who cannot be targeted, so the treatment is really not working here at all. But this one, this is not the primary population. These are actually reversibly exhausted cells, the Y2s, the red ones, are the ones that are dominating in the protocol where actually things worked. Interesting. So the next thing also we wanted to look at is this inflammation that we are terming this. Again, we're not talking about any specific cytokines at this point. We might get a little bit more specific or hypothesize about some specific things to look at a little bit later. But right now, kind of more local inflammation. And you can see here, again, this intuitively makes sense, but it is still helpful to see that. So this is a failed protocol and this is failed protocol, and your inflammation's really high. So above this, my threshold F2 of transition to terminal exhaustion, which again makes sense. Why then all of your, or primary majority of your immune cells will be in this terminally exhausted state, because that's what the system dictates it to be. It says it's too much going on. But this successful protocol, my simulated inflammation stays under this threshold, or maybe taps it a little bit but goes down. Interesting. Maybe there's something here. So then we wanted to then look more closely at this inflammation idea and how much do you always have to stay under? Is it really like all about that? So we calculated how this accumulated inflammation, so we did the area under the curve, essentially, between this level F2 and however high it gets. And we saw how much of my simulated inflammation went above this threshold. And so you can see, so these are the region of kind of successful treatment and region here. This is yellow is really high, blue is super low, and this is kind of intermediate. And so what we're seeing here is that responders have inflammation levels that either do not cross it, like all these blue ones, or transiently cross this threshold. So it is kept under, like within some range that prevents transition to terminal exhaustion. Again, makes perfect sense intuitively, but it's still really helpful to see. Okay, so that's very nice. This is theory. Is this anything that actually can be seen in patients? So this was a kind of an extension project, and I have a separate story focusing on this work that I'm going to briefly touch on here. But for now, I'm just going to mention so that we analyzed, like in a collaboration with a postdoc, Clara Pavie, that we looked at published single-cell RNA sequencing data set. And these contained samples collected during phase 2 clinical trials. We had 27 head and neck squamous cell carcinoma patients that were treated with either just anti-PD-1 or anti-PD-1 combination with anti-CTLA-4. Just another checkpoint inhibitor, targets a different checkpoint, but same idea. We identified the gene signatures that were associated with different exhaustion programs, because we have to look at the cells and say, okay, based on your gene signatures, who are you? Are you terminally exhausted? Are you reversibly exhausted, immune effector cell. So Clara put together this gene signature data set to identify this, the cell state and the cell program. And then we looked at these data to ask two questions. One is, are there differences between responders and non-responders when it comes to different states of exhaustion? We're predicting it, and it is intuitive that this would be the case, but do we actually see it? And the second question that on some level could be even more interesting is an intrinsic property of a patient, or is it something that is mitigated by treatment? And again, I have a hypothesis and intuition based on the model and on kind of understanding of this, but of course we have to actually check. Well, so snapshot of results. First, we looked at post-treatment responders versus non-responders. And you can see from the data, so the blue ones are the terminally exhausted cells. Responders have fewer of those compared to non-responders. Non-responders have more terminally exhausted cells. Yes, we are predicting it, and it makes perfect sense intuitively, but it is really nice to see this in data. There was also, unfortunately, a very small data set. There were only 6 patients for pre-treatment versus post-treatment to see if this is something that is mitigated by treatment versus not. In a perfect world, we would see pre-treatment and post-treatment for responders versus non-responders. We couldn't split that here because it was a really small sample size, so they're pooled here. Nevertheless, there is a difference in immune cell composition before and after treatment. And particularly notable, again, the blue ones are my terminally exhausted cells. They are higher in post-treatment. Interesting, which again makes sense if the treatment is causing some immune activity. That would cause some inflammation to accumulate. Some of that might trigger transitions to reversible and then terminal exhaustion, which would increase these proportions relative to pre-treatment. Okay, interesting. So treatment can change composition of T cells with respect to exhaustion, and it's not necessarily an intrinsic patient characteristic, which is a great thing because that means that something can be done. It's not just dumb luck that, oh, oops, I'm just not a responder. Maybe there's something you can do with it because this is such a fundamental biological program that really evolved for dealing with a different situation with infections. And so everybody should have some variability there and some system for dealing with this. Okay, so at this stage, we're asking, we identified some protocols. Is there a strategy that is more likely to work for more individuals? Like we're showing that these 3 general strategies, either having this MTD-like, so high dose, low frequency, or this intermediate one, or the low dose, high frequency one. So any of these can work. But is there one that is more likely to work for more people? That is the question. Okay, so how did we approach this? This was a time when we were just starting with this idea. We didn't even name them virtual populations at the time when we were working on this, about 2 years ago, I think by now, or maybe more. Well, but nevertheless, the idea is the same. We assume that every individual has their own value of, we just selected one parameter here. Now when we do virtual patients, we do more, but here F2. So this threshold of transition. And assuming that for some people, maybe it's going to be maybe over here, and then they get to terminal exhaustion real quickly. And maybe some of them are over here, and they are going to be responders regardless of anything. I don't know. Or maybe their inflammation accumulates differently, but that's a different topic. So looking at where, like assuming that each person might have a different value of F2. So then we create this population of individuals, but they're really, all the other parameters are kept the same, kind of average, and each person just has this different threshold. And we run this dose schedule sweep that I was talking about. And then for each combination of doses and schedules, we ask, did the protocol result in tumor elimination? And then we calculate the proportion of individuals for whom this protocol worked to see which protocols would be most likely to work for different people or for majority of people. Okay, so then we get results that look kind of like this. And here the yellow color is when everybody responded. So we are seeing very clear patterns that. So this is small doses, because you divide 42 into many, many pieces. Small doses given far apart do not work, kind of makes sense, probably do not just trigger enough response. So large doses given too close together also don't work, probably cause too much inflammation and lots of issues right away, do not even get to the response. And then there is this region of yellowness. And over here, we are actually seeing that just based on this, the protocols that are this third type of strategy, kind of low dose, high frequency, is, according to this, within the frameworks of this biological system, under these assumptions, is more likely to work for more individuals. If given, of course, as monotherapy. We're not looking at combinations yet. Okay, that's interesting. Just for curiosity, we also looked at the impact as cumulative dose increases. There we chose like a small number, 40, so like 40, 50, 60, 70. So for higher doses, if you have higher doses to subdivide, of course, the range of protocols that are successful for more individuals increases because you're essentially given larger doses. Okay, but this one also, you're starting this kind of smaller, okay, larger and larger, but it is still this kind of diagonal. And MTD-like protocols can be effective for large cumulative doses, which seems like is where we ended up these days, but like less so still than if there was more exploration in here. Okay. Before I go to the next thing, I just wanted to address one question. This actually came from the reviewer. It was an excellent question. We are analyzing this, operating under the assumption that it is inflammation, some kind of systemic or local, most likely local inflammatory markers that are triggering this transition to exhaustion. What if the trigger for transition is actually chronic antigen stimulation, which is another kind of very studied and big hypothesis and proposed mechanism. John Ware's group have been studying that a lot. Well, at this stage, it's not even which one is it? Oh, is it this one or that one? For practical purposes, the question is, does it make a difference to predictions? So this is what we had for our regular, our inflammation-driven transitions. But then we modified the model to include this mechanism of transition between exhaustion states due to chronic antigen stimulation instead of inflammatory signaling. And then we reran the whole thing. And the results are qualitatively the same. So this here is, we made sure that we're comparing apples to apples, that the trajectories without treatment are the same, so our simulated tumors are essentially identical. And then when we ran this, actually, qualitatively, the same, not quantitatively, but qualitatively, they're largely the same. And in fact, kind of this inflammation-driven hypothesis is more conservative. So there are fewer schedules that we are predicting that would work for majority compared to this one. This one is much more optimistic. As whenever I do dose projections for my work, I personally am a little bit more conservative. I would rather be pleasantly surprised than plan for too good an outcome. So if I were taking this and parameterizing to something real and then working with it, then I would stick to the, frankly, more conservative hypothesis. But we can at least now see that qualitatively, this assumption does not make a difference. Okay, so has anyone actually tested this? Again, we find this in the data, but now we're making a prediction about protocols and changes and all of these things. Well, when we were looking at this, at the time when we were looking, there actually were not that many or any trials looking at de-escalation. Right now, I just checked, there are some, mostly looking for non-inferiority outcomes, so reduce the dose, but mostly looking at the dose, adjusting the dose, and then seeing if the outcome is about the same. Because typical protocols involve administration of high doses every several weeks, usually every 3 weeks. Some are looking now at, can you space it out more to every 6 weeks? Some are looking at, can you give a lower dose, like instead of 400 milligrams, for example, give 200 milligrams at the regular schedule. But still the outcomes that they're looking at are mostly non-inferiority. But there are several retrospective analyses that were done for patients that were given lower than recommended doses of either the ones that I found at the time, Pembro or nivolumab, both are PD-1 checkpoint inhibitors, because availability, financial reasons, things like that. And it was very interesting to see that a lot of these studies showed that lower doses can be at least as efficacious as standard of care, and obviously often with fewer side effects. So that was pretty cool. So it seems like we're kind of fishing in this space, and that's okay, that's already interesting. But of course, more frequent dosing can pose a lot of logistical challenges. These molecules usually have really long half-lives. That is a very wonderful feature of them because you do not need to use them, like come for them very frequently. But, they require an IV or like a hospital visit. You can just easily do them, and that logistically is just really hard to do. So it makes a lot of sense that reducing the timing between doses is not something that is going to be working very well. So the question is, well, maybe there's something that can be done in terms of adding a combination partner to mitigate something like this. So we wanted to look at it again. This was a reviewer comment. And we were like, that's an excellent question. So we did sensitivity analysis on our system. And the way that we designed it is, again, what's the question? Why do we care about the sensitivity here? Or sensitivity to what? We wanted to know what mechanism should we target to flip the outcome of therapy. So what we want, but not all of them. We want to turn these red regions green, but we want to keep the green regions from turning red. Which mechanism in our system can we target in order to achieve that? The way that it was done, so we were varying each parameter, and if we varied it by 100% and still didn't flip the outcome, we designated this as an insensitive parameter. And the goal was to find parameters that can, as I said, flip ineffective protocols to effective, but not to flip effective to ineffective. Before I go through the results, just kind of example of the output. So, for example, if something is all blue, this is something that is sensitive across all protocol space. And that would mean that, yes, it might flip ineffective protocols to effective, but it will also flip effective into non-effective. We don't want that. If it is yellow, then it is insensitive across anything, so it does not affect anything. What we want to see is something that looks like this, which flips different regions. So this one, for example, would be sensitive. So the red one here would be sensitive across protocol space. We would want to flip this, but we want it to be insensitive in this space. Great. Okay, so now what are the results? So we varied all of the parameters, and you can see already that, okay, so these are all way too sensitive, and there are some that are completely insensitive, but there are some interesting ones that we're going to talk through in a moment. Okay, so these parameters here, they relate to killing capacity of terminally exhausted T cells, how well they can kill tumor cells. Don't think that there is a drug that can actually affect that. So that's probably, while this is a sensitive parameter, probably not something that we can mitigate pharmacologically. These ones, okay, so this one, the C1, was rate of cytotoxic T cell expansion due to tumor kill. I mean, maybe something you can, maybe CTLA-4s, now that I'm thinking about it, could be an option here. Don't know. But might be kind of hard to control this, or no, not CTLA-4s, never mind. Or the death rate of exhausted T cells, again, how quickly they die. Is there something that we can do to delay that? Probably not. But then there's this guy, inflammation-driven transition to terminal exhaustion. So that is a parameter that was kind of associated with that term, which featured the F2 threshold. And that may be something we can do with. And so the proposal would be here to do a combination partner that actually, counterintuitively, could be an anti-inflammatory drug. Well, funnily enough, 6 months after our paper came out, there were 2 separate clinical trials that were published that found that a combination of immune checkpoint inhibitor with this particular anti-inflammatory drug, it's a JAK inhibitor, dealt with interferon gamma's reduce the inflammation based on this marker, can actually improve the effectiveness of checkpoint inhibitors and even rescue response. In fact, and it was shown for both solid and liquid tumors. which was very interesting. So this one was metastatic non-small cell lung cancer. And as a monotherapy, under 50% response rate, high recurrence, but combination with a JAK inhibitor, 67% response rate and much lower recurrence rate, median recurrence rate over 2 years. That is remarkable. This one was in Hodgkin lymphoma, and actually the inclusion criteria was that they failed monotherapy as immune checkpoint inhibitor monotherapy. But when combined with a JAK inhibitor, 53% response rate, including 46% of patients showing no progression after 2 years. I mean, this is absolutely remarkable. But one important thing here was that it was essential to appropriately time the anti-inflammatory drug, not to give them together at the same time, which again, thinking through this intuitively makes sense. If you are, the inflammation here serves a purpose and some of it will create a larger pool of your kind of reversibly exhausted cells that you can target with a drug. So giving it together, then you actually pre-dampen your immune response. But if you let it do its work, it now is starting to get to the transition space of, oh no, we're getting to terminal exhaustion, so crossing the threshold of 2, adding an anti-inflammatory to tamp it down would actually keep your immune cells in that targetable state. Again, intuitively makes a lot of sense. Computationally is how we probably figure out schedules, but this was really, really exciting to see. And so kind of bigger picture is if we're trying to think through this whole, we have this hot, cold tumor paradigm, that cold tumors are not responsive and hot tumors can be targeted by the immune system. So in a cold tumor, maybe like lack of responses due to insufficient immune stimulation. And in a kind of a warm or hot tumor, maybe you get the response, you have sufficient immune stimulation, and this is when checkpoint inhibitors can work. But we're kind of suggesting that maybe this is actually a little bit more of a bell-shaped curve, where you can have too hot a tumor, which is also not responsive because you have so much immune stimulation that it actually triggers this protective mechanism against autoimmunity. Transition to terminal, irreversible exhaustion that is not targetable. And so this idea that it's not binary, you can have lack of response for different reasons. Maybe it's insufficient stimulation, or maybe there's too much fire going on there, and that is something that maybe should be mitigated with something like an anti-inflammatory. Okay, this is what I got right now for this story. This is the paper that came out. So this is the model is in the this NPJ Systems Biology publication. And then this is the work that we did, as I mentioned, with Clara Pavier on analyzing patient data and kind of really single cell RNA-seq data. And I have a separate story, kind of a second part to this, where I'm diving into the details of what we found in here. So please stay tuned. Thank you for your attention.